Hello. Brain Health Alliance and the BHAVI Institute is very happy to present this talk by Sarah Banks, PhD. She is a neuropsychologist on faculty at UCSD, where she treats patients with neurological dementias and neurodegeneration. And I am thrilled that she's going to be able to give this talk for you. Please go ahead, Sarah.
Thanks so much, Julie. I appreciate it. So today I'm going to be speaking about Alzheimer's disease and specifically sex differences in risk and expression. So to give you a framework for today's talk, I'm going to briefly define what I mean by Alzheimer's disease. Of course, most of you probably know, but just to give the context specific to how I see it and how I view it in my research. I'll then talk a bit about how sex differences can help us to answer important questions about Alzheimer's, what factors might promote Alzheimer's in women specifically, and then how we might be able to help people to reduce their risk of cognitive decline with aging. So I wanted to take a moment just to provide a big caveat, recognize this caveat for today's talk. So when I talk about sex differences, I generally oversimplify and talk about men and women, males and females. This is an oversimplification, and I think we need to do a lot more to incorporate the breadth of gender factors and the complexity of sex differences. But I just wanted to provide that piece of information and insight today, just to say there is more to it than that sort of binary division. So what is Alzheimer's disease? It's a brain disease, of course, and is the most common form of dementia. And by dementia, I mean the progressive loss of cognitive abilities and independence due to brain disease. So while Alzheimer's disease is the most common form, it's certainly not the only form. And there are other primary types of dementia, such as vascular dementia, frontotemporal dementia, Lewy body dementia, and then most people have some mixture of those different diseases in their brain by the time they die of a dementia. It's driving my research is the prevalence of Alzheimer's disease. So 1 in 3 of our older adults dies with Alzheimer's or another dementia. More than 6 million people are currently living in the States with Alzheimer's disease. And these are facts and figures from the Alzheimer's Association. Importantly, nearly 2/3 of these individuals, these Americans living with Alzheimer's and dementia, are women. So it definitely doesn't affect people equally, whether you're a man or a woman. And it's not just because women live longer. So what does Alzheimer's disease do to the brain? So this little cartoon shows 2 sides of a brain. In one side, the left side, we see this nice plump cortex where there's minimal atrophy. And then we see 3 different cartoons of different cells that we find in the brain, neurons or brain cells, astrocytes, and microglia. All 3 of these different types of cells are impacted differently by Alzheimer's disease. So in between the neurons, we see amyloid beta plaques building up, these sticky, starchy material that lays down in between the cells very early in the disease process. About 10 or 15 years after amyloid has become diffuse within the brain, we start to see neurofibrillary tangles forming. And they happen inside the neurons. And microgliosis and astrogliosis are important components of the spread of the disease that are part of the brain's normal immune and inflammatory response, but seem to go awry specifically in Alzheimer's disease and other dementias. The timing of some of these changes is important, especially when we think about interventions. So as I mentioned in the previous slide, about 10 to 15 years before we see significant clinical decline, we have a buildup of amyloid throughout the brain. At some point during that process, tau, or those neurofibrillary tangles, begin to spread, and those predict very closely what kind of cognitive symptoms we're going to see and when those are going to occur. So typically in Alzheimer's disease, the first cognitive changes affect the memory systems. And that's because for some reason, tau has an affection for spreading in the entorhinal and medial temporal lobe, which are important in memory. Eventually, we see a breakdown in clinical function and ability for the individual to be independent. And that's where we go from mild cognitive impairment, where problems are focused on cognition and not really interfering with independence, into the dementia phase of the disease, where people begin to need more and more help to get by day to day. I'm going to zoom in a little bit on tau pathology because it's my favorite pathology in Alzheimer's disease, partly because it's linked so closely to cognition, which is my day-to-day job. So tau is thought to have a healthy form, or we know it has a healthy form, and we think that that stabilizes microtubules. However, when there's this breakdown associated with Alzheimer's disease, tau oligomerizes and builds up in cells, and it likely propagates cell to cell, explaining the spread of the disease. Tau tangles build up in the cell body as these intracellular neurofibrillary tangles, which prevent the normal functioning of the cell. And this leads ultimately to cell death and atrophy. And all that's left eventually in the cell is the extracellular neurofibrillary tangle, or a ghost tangle. So there's a process with tau pathology that hopefully we'll find a way to interrupt. So amyloid has this diffuse spread throughout the brain, and it doesn't really correlate very much with cognitive dysfunction. Tau, however, is very, very strongly predictive of cognitive decline. In many middle-aged adults, we'll see some degree of tau, very much limited to these early stages that neuropathologically have been defined by Braak and Braak as stages 1 and 2. Generally, only in the accompaniment of widespread amyloid, we see a spread of that pathology into stages 3 and 4, which you can see here encapsulating more of the temporal lobe, a little of the occipital lobe, a little of the parietal lobe. And that's generally when we see mild cognitive impairment. Eventually, of course, the disease spreads throughout the brain, and that correlates with the timing of dementia. How do we measure tau? Well, the gold standard for many, many decades was autopsy, or the only way really of assessing tau was at autopsy. So for most people, we didn't know that they definitely had Alzheimer's disease until they were dead. However, we can tell in cerebrospinal fluid, which of course gives us a pretty direct idea of what's going on in the brain. And now, excitingly, we can use PET ligands to tag and visualize not only how much tau tangles there are in the brain, but where they are. And even more excitingly, perhaps more recently, there's a hint that we can measure amyloid and tau in the blood, which is really going to change the way we can do Alzheimer's research. I use tau PET a lot in my studies, which of course is expensive and complicated, but it does have that advantage of being a fairly direct measure of tau and showing us again where the tau is in the brain. So earlier I mentioned microglia and astrocytes and mentioned that they have an important role in the process as well. And the thought is that while microglia and astrocytes might have an important role early in attempting to sort of flush out the oligomers and the bad proteins as they form in Alzheimer's disease, eventually that process might go somewhat awry and actually create this detrimental loop, which accounts for more and more tau spreading. So it's thought that inflammation is a really important factor in the spread of tau. And again, potentially a little window into something we might be able to change to reduce spread or perhaps stop the disease. Sadly, as most people know, most treatments haven't worked. On the left here, this is the Tau pipeline. This one's a couple of years old, from 2022. Jeff Cummings puts out this pipeline, which is a synopsis of all of the different drugs and their targets each year. And here you'll see the breadth of different kinds of drugs that we have, from disease modifiers to cognitive enhancers. And in the different colors there, we see different kinds of targets that they might have, and in the different rings, the different stages of the drug development. Each of these drug studies costs millions, if not billions of dollars, and they can take a decade. Sadly, to date, there's been a higher than 99% failure rate of these drug studies. Of course, there have been recent breakthroughs, but they've in drugs and how they impact Alzheimer's disease, which we'll talk about further. But there's also a different kind of hope, and that's with new understanding of modifiable risk. So in 2020, the Lancet Commission brought out a paper where they determined that more than 40% of risk is modifiable. And they broke this down in a meta-analysis of the literature as early, midlife, and late life risk factors. So you can see on this sort of reverse S-curve on the side in this diagram that there's education early in life in green there, and then things like hearing loss and hypertension in blue, and in later life, smoking, social isolation, physical inactivity are all important predictors and modifiable risk factors for Alzheimer's disease. So this is really important. A lot of these things are things that we have really effective treatments for. Sometimes they're behavioral, they require behavioral change, which is very hard for people. But it's exciting and it offers an avenue. They haven't done a sex stratified Lancet Commission. It would be really interesting to see that, because we know that many of these factors have sex or gender effects. Very recently, just at our annual meeting a couple of months ago in Philadelphia, the Alzheimer's Association International Conference, this group shared their update where they added cholesterol and visual loss as two important modifiable risk factors. They still haven't included sleep in this, which they explained as being because we don't have a very solid understanding of direction. But I think sleep is one that many of us would agree is a very important risk factor too. So it's gone up now. It's at about 45% of risk is modifiable. So the more we understand, the higher the number seems to get. I would argue that sex stratification might be able to help us find more effective treatments. And that's because of sex differences that I'm going to move on to talking about next. So that was all the sort of background about what Alzheimer's is, and I now want to touch on 3 research questions. One about sex differences and the risk and expression of Alzheimer's disease, another on what factors promote Alzheimer's in women, and finally, I just want to touch on how we might be able to help individuals to reduce their risk. So I'll start with, excuse me, don't look. I'll start properly now with sex differences in the risk of expression of Alzheimer's disease. So Alzheimer's, of course, doesn't happen suddenly and without context. It happens in the context of our lifespans. And throughout our lives, men have a slight visuospatial advantage and women have a verbal advantage. So just to walk you through these graphs quickly, in the top left, we have visuospatial ability, and men are in the solid black line there and women are in the dotted line. And whether we look at adolescence, young adulthood, or older adulthood, there is a slight advantage for men over women. If we look at visuospatial memory specifically, in the bottom there, we see less of a difference, but there's still a slight advantage for men. Women, on the other hand, show a solid advantage, the dotted line in the top right, on verbal ability throughout the lifespan. And again, at the bottom, verbal memory is different throughout the lifespan. So this is a blessing and a curse. Of course, it's nice to be good at something, But of course, the way we test memory generally in clinical trials, especially, but in many studies of Alzheimer's disease, is with verbal material. We use memory lists like this one, which we might read to a participant, and then ask them to recall as many as they can, and then do it again so that they build up a nice learning curve, and then put it away, and 20, 30 minutes later, spring a recall on them and ask them, oh, you remember that list I read to you earlier? Can you please tell me as many of those words as you can now? And that will give us an idea of their ability to learn and also later recall and retrieve that information. Purely verbal, of course. So a woman doing badly on this test, for example, at a specific cutoff, will probably have more amyloid and tau in their brain than would a man at the same cutoff. And yet we very, very rarely have sex-specific cutoffs on these tests. And we see that when we look at data. So this is a study by my colleague and friend Erin Sunderman, who's now here at UCSD. And she looked at the Alzheimer's disease neuroimaging data, and which accounts for individuals with normal cognition, those with mild cognitive impairment, and those with dementia. And what you can see here is the women in blue and the men in the sort of pinkish salmon color. And the women on this verbal memory task, when we look at the whole sample in the top left, do better as a whole. And they decline somewhat as a function of cortical amyloid. When we look at normal cognition, the women have an almost parallel line with the men, so they're not declining much. There's not as much amyloid for the most of them in the brain, but there's a solid advantage there, despite the amyloid in the brain. As we move into MCI in the bottom left, we see, again, a bit more decline, but the women maintaining that advantage. And then once we get to the dementia phase in the bottom right, the advantage disappears. So women kind of fall off a cliff a little bit and decline more rapidly at the end, down to the level of their male peers. So we see this verbal memory advantage, but we do think that it contributes to underestimating AD pathology in women versus men. Sex differences in cognition can also impact trial design. So this is a study we put out a few years ago looking at cognitive composites. So when we kind of smush together a bunch of cognitive tests to measure an outcome. And this is a very popular technique in clinical trials these days. So not relying on a single test, but looking at a few tests and creating these composites. And what we did is we created data-driven composites based on what women are good at and what men are good at. And we created these lines on the curve so that using the sex-specific composites, you can see that we have a much, I'm sorry, I should explain this a little bit differently. This is a power curve. So this shows us how many people we would need in a two-armed study if we wanted the different power levels on the left side. So if we look at a power level of 0.8 there, and I think you can see, yes, which is generally what we want. If we use the sex-specific composites, we need about 250 people in each of our arms. But if we use a typical published composite like the ADNI-PAC, which is used very, very often in studies, we're going to need hundreds and hundreds more participants. We're going to be off this chart into probably the 600s or 700s compared to these sex-specific composites. So taking into account lifelong differences in cognition would be very advantageous in the study of clinical trials. Excuse me. So, I'm sorry, my slides aren't advancing. There we are. Oh, they're advancing at my end, but they're not advancing on the screen. There we go. Now it's working again. So sex differences in cognition can also be seen in different levels of tau. So what we see here is a study based on the A4 data, and A4 is a clinical trial which used people who were cognitively normal, and they used some very interesting cognitive measures, including some computerized cognitive testing and some really exciting technologically advanced cognitive tests. And what we can see here is men in blue and women in red. And with this kind of graph, if the line is crossing the vertical line in the middle, the data is not significant. But if the line doesn't cross that, then there is a significant relationship. And what we can see is that for many of these tests, and I don't have time to go through the specific details right now, we see that women show significant relationships between the cognitive tests, these are very sensitive cognitive tests, and tau, whereas women do not show those relationships. So if I was designing a study of early Alzheimer's disease, these are people who are amyloid positive but not yet having cognitive decline, I might use some of these measures, but I would like to know that they are significant in women and not men. And there's this one study, this one test here, which was significant in men but not women. So important sex differences in cognition could be a really important way of incorporating sex differences and understanding more about early processes in Alzheimer's disease. In addition to the differences in cognition, women actually accumulate more tau than do men. So for some reason, during the MCI phase especially, women have kind of this inferno of tau in their brain. And we saw this on this PET study, again with the Alzheimer's Disease Neuroimaging Initiative data, where we sex stratified amyloid positive individuals with a diagnosis of MCI. And what we saw is that the women show much higher levels of tau and the brighter yellow color here than do men. The pattern of tau distribution is the same in men and women, and it's what we expect based on those Braak stages that I showed you earlier. But there's just a more tau in women than there is in men. So again, why is that? And what can we do about it? Despite having higher levels of tau, women continue to do better on verbal memory tasks. And this is some data that was presented by Dino Digma, former RA in my lab, who's now in his neurology residency. And what he showed is that in whether we look at people when they're still alive using tau PET, women who are in red in these diagrams have a higher level of verbal memory reserve than do men who are in blue. We can see that on this scatter plot where there's more red dots above the black line than there are blue dots, or we can see it on this cello plot where we see a fatter top to the women's plot than we do with the men. We also see it if we look at autopsy data. So this is data from the National Alzheimer's Coordinating Center, where data from all over the country is combined into a database. And we can see that whether we look very early in the disease process or in that sort of MCI type phase, we see that these this heavier distribution towards the top of the verbal memory ability in women in red compared to men, whereas just like in Dr. Sunderman's research that we saw earlier, once we get to the dementia phase, the later phases of the disease, there's no difference and women lose that reserve. So what Dino thought there is that women could really bear this bigger burden of tau. So we have more tau in women's brains, but still doing better verbally despite that. I'm going to skip over this one just in the interest of time. We also see sex differences in some of the biomarker measures. So this is a study by Amaryllis Tsiknia, who was in my lab. She's now doing her PhD at USC. And she looked at how plasma p-tau181, which is taken from a blood test, correlates with tau PET in the brain in the entorhinal region here, which is very important in early Alzheimer's disease. And what she saw is that women actually in purple here had a much stronger relationship, very highly statistically significant, between their entorhinal tau and these plasma levels. And we see that as well if we look instead of tau PET at CSF PET, so another kind of direct measure of central nervous system pathology. And what we see is, especially during the MCI phase, here again, women have a much stronger relationship between the level of CSF tau and the peripheral measurement from the blood. And that's important as we move into using blood tests more frequently. It's really important that we understand what they're measuring and that there might be differences between men and women in these relationships between peripheral and central values. A potential mechanism for that is the permeability of the blood-brain barrier. So of course, if we want to take blood to determine how much tau, other pathology there is in the brain, we need to understand what's getting through the blood-brain barrier, because that's how it gets into the peripheral system. And what we see is if we look at MMP9, which is a endopeptidase, which is mediated by estrogen and regulates blood-brain barrier permeability, what we see is a stronger relationship between MMP9 levels in the plasma and tau levels in the central nervous system with CSF in women, again in purple, compared with in men. So that's really important. We also see that the MMP9 levels, so this assay of permeability of the blood-brain barrier, is more significantly related to cognition in women than it is in men. So there seems to be something there where maybe blood-brain barrier permeability is different in men and women, and that might be impacting some of these plasma measures. So of course, one of the differences between the sexes is chromosomes, and chromosomes are genetic matter. So what about genetics? Do they differ in how we understand sex differences in Alzheimer's disease? Well, they do. So the most highly predictive genetic risk factor that we know of, at least in the white people who have been studied more thoroughly than other race and ethnicities, is that ApoE4, apolipoprotein E4 alleles, of which you can have one or two or none, are a strong predictor of whether or not you develop Alzheimer's disease later. So Amaryllis here again, when she was working in my lab, looked at how ApoE4 and sex modified the relationship between cardiovascular risk, which is again something with a lot of sex differences, and cortical tau. This is looking at the Alzheimer's disease neuroimaging data once more, women in red, men in blue. This is the Framium cardiovascular disease risk data. So it's the ADNI data, but using the Framium risk score. And what we see, even though these people in ADNI, the participants, were pretty healthy in general, even a little bit of vascular risk, as measured by the Framium risk score, correlated with a much higher level of tau in the entorhinal cortex, inferior temporal, or a composite temporal cortical region. All of these are very important in Alzheimer's disease. In women, in red, but not in men, in blue. So even a small amount of vascular risks, if you're a woman and you're an E4 carrier, seems to be very problematic in terms of your tau load. So I think that's something that's very important when we start thinking about precision medicine and ways to intervene. So women and men differ in other genetic risk factors too, beyond ApoE4. So we look at something called a polygenic hazard score, which combines numerous different genetic risk components and also adds, beyond just a polygenic risk score, of which there are many, the polygenic hazard score also takes into account time, which of course is very important in a disease like Alzheimer's disease, where aging is the number one risk factor. So polygenic hazard scores take into account this polygenic risk plus age. Typically, they are not sex differentiated. You get one score whether you're a man or a woman. We did something a little bit different where we created sex-specific scores. And then we calculated an individual's risk based on their sex-specific score, or the opposite, like we used male data and we applied the female score. And what we see is if we use the sex-matched scores, we get a very, very significant prediction of their AD onset, of their AD onset with pathology confirmed, or their annual changes on cognition and function. And then it's a little bit better with the sex mismatched, but if we use the risk scores which don't involve age in any way, there's much less of a prediction of these sort of when someone's going to decline. So adding that sex-specific component really boosts the significance of these scores. If we break down these non-ApoE4 sex-specific risk factors, and then we apply it particularly to tau, since tau is so different between men and women, perhaps unsurprisingly, we see some specific relationships between some of these genetic factors and tau in women. So the difference between the green curve and the pink curve shows this sex-specific additional risk in women and not men. And these graphs show normal controls, MCIs, and dementia participants. And what we see is that most of the changes are in this middle section, where people have more BRAG in their 3-4 regions, is consistent with MCI, which is, again, when we see that kind of inferno of tau in women. Why does this all matter? Why should we care about this? Well, this is data that was produced by the company who make Leqembi or lecanemab, which is now an FDA-approved drug which removes amyloid from people's brains. And that's a great idea, right? You don't want amyloid in your brain because it's associated with Alzheimer's disease. But there's significant risk factors. People have died as a result of the side effects, and there's also a lot of expense that goes into this. And the payoff in the combined group is a slowing of cognitive decline by about 4 months. So people get about a 4-month window of improvement, and that's in combined men and women. So when they designed this study, they created about 50% women and 50% men in the cohort, which of course does not reflect what we see in the population, where it's 2/3 female and 1/3 male. But I really credit the company with sharing this sex-specific data, where you can see, and again, in these graphs, if the horizontal line touches the vertical line, the analysis is not significant. When we look at the women in the study, they did not show any improvement as a result of using this drug. And this was true of all of their primary outcomes. This is a purely cognitive one. This is an activities of daily living. We see an improvement in men at the bottom there, but in the top here for women, the lines are crossing or touching the middle, which suggests that there's no difference. So, to me, that is a really significant and important piece of information that should be shared more widely. So what factors promote AD in women, and why should we look at that? Well, myself and my colleague Erin Sunderman created the Women, Inflammation and Tau study because we were very interested in this idea that women have higher levels of tau in their brain, and they also tend to be more vulnerable to inflammation. So we thought that maybe inflammation promoted tau in women, and modifiable risk factors such as sleep apnea, sedentary behavior, or poor diet might promote inflammation, and we think that that might cause an increase in tau in women compared with men. Because there are so many differences in these different risk factors and also how Alzheimer's expresses, we chose to focus our energy on women specifically. We were funded initially by the California Department of Public Health and now the National Institute on Aging. And we're looking for 110 women aged 65 and older with an elevated risk of Alzheimer's based on their polygenic hazard score and a MoCA range suggesting some level of cognitive loss. So we call it MCI-ish. We've recruited primarily through geo-targeted mail-outs and now from community events. We do a screening visit, which involves a MoCA now over the phone and the shipment of a saliva kit, which they ship back, and we do the DNA testing. And then they come in, and these women are really fantastic. They provide a huge amount of time and body fluids and sweat and brain pictures with MRIs and PET scans, blood draws, lumbar punctures, sleep studies, physical activity, just a plethora of different measures. We have about 50 women who are in the study already. This is just another depiction of what they go through, and about 20 of those women now have come back. So we are getting some preliminary data, which I'll show you a little bit of in a minute. This is one of the studies that we used as an early basis for why we did what we're doing, showing that women, I can just show you this box here, women in dark gray having a much stronger relationship between this inflammatory factor and cognition compared to men in light gray. This was Rachel's work. Rachel Bernier was our first postdoc on the study. So women show this stronger relationship between inflammation and cognition in aging. And the participants in this particular study were controls, normal, healthy controls, as well as MCI and Alzheimer's disease. And this is the model that we're trying to test. So we're trying to determine whether modifiable factors, specifically exercise, insulin resistance, and sleep apnea, predict Alzheimer's outcomes, specifically pathological tau and cognition, and whether that's mediated by some of these inflammatory markers, specifically TNF-alpha and IL-6. And we also look at sex hormones, including testosterone. Because we know, of course, that verbal memory is not the optimal way to assess memory in women due to their verbal memory reserve, we make sure that we also focus on nonverbal memory. So we give a test like this, the brief visuospatial memory test, where we show the women in the study an array of shapes such as this one. They see it for 10 seconds and then it's taken away. They're then asked to draw it from memory. And they do that 3 times so that they can learn it, and then again later. When we look at the data from the brief visuospatial memory test in other studies, we don't see any sex difference. So we think that this is a kind of sex-blind study, and it seems to be particularly significant in relation to AD outcomes in women. So this is some very early data that we shared at a conference from the study, where we showed that the brief visuospatial memory test was a much stronger predictor of tau than was the verbal memory task, probably because of the noise in the verbal memory task caused by some women having that strong reserve. Something else that we're seeing that's really motivating for us to keep going with the study is a very strong relationship between sleep apnea and different sleep metrics and tau. And also a kind of shocking statistic that over 70% of women in the study, despite not knowing they had sleep apnea, have sleep apnea when we test for it. And that's in part because sleep apnea expresses differently in women and men, and most clinicians aren't aware of that and don't sort of screen for it in women. So another modifiable risk factor. under-recognized in women. Kitty Liu, who's a graduate student on the study here, is looking at various aspects of sleep. The data I'm sharing here is simply sleep complaints. So a brief questionnaire, the Pittsburgh Sleep Quality Inventory. When we give it to women, the women in the study, they have a very strong relationship between how well they think that they're sleeping and the tau in their brain. And similarly, we see in the middle here a strong relationship between sleep quality and memory, but only when we use that nonverbal memory test. So showing how important it is to factor in sex differences in cognition. And again, we see a strong relationship between tau and that nonverbal memory test, as Alex showed on the previous slide. So we have this fabulous study that we love, Women Information and Tau Study, or WITS. And Erin and I have been trying to figure out how to diversify the study. So right now, most of the women in the study are white, non-Hispanic, not all, but most. At the same time that we've been kind of struggling with this, the technology in blood-based biomarkers of Alzheimer's disease has really kind of exploded. We're in a great time in the science now for that. And what we decided was that rather than try to have maybe 10–15% African American participants in the study, we would focus an entire study on that cohort. So I'm going to talk about that in a second. And part of the motivating factor for that, beyond just wanting to have a better understanding in non-white individuals, is that Black African Americans are at the most at-risk group in the country. So for every, if we look at adults over 65, 14% of African Americans in that age category have Alzheimer's disease or another dementia, whereas 12% of Hispanics, again a big number, and 10% of non-Hispanic whites have Alzheimer's disease. So for some reason, it's stronger in Black or African American individuals, and we don't know how that interacts with sex, but we expect that Black women are even at higher risk. And why is that important? Well, Black individuals have been historically underrepresented in science and in Alzheimer's disease clinical trials especially. And you can see from, again, the lecanemab data that we had hundreds of white individuals, 860, and only 40 Black individuals. And that really is difficult when we try and look at the race stratified data, which proves completely insignificant, although it's a very, very underpowered analysis. We really need to know more about this very at-risk group. So BWITS is the Black Women Inflammation and Tau Study, which has recently been funded. And this allows us to leverage that technology in blood or plasma markers and bring WITS into the community. We're recruiting using community-based participatory research practices. So we spent the first year of the grant really understanding what we could do to make sure that this study is done right and is done in a way that's beneficial to the people that we're studying. We're working very closely with Dr. April Thames, a co-investigator on the study who's based at UCLA. She'll be recruiting 50 of the participants, and we'll be recruiting 50 here in San Diego with Dr. Mark Norman, who grew up here and is very well connected with the local African American Black community. He's also an expert in race as it applies to neuropsychology. We removed several factors from WITS for BWITS. Of course, any woman is welcome to be in WITS if they meet the criteria. But for BWITS specifically, we removed the imaging and the lumbar puncture, and we also removed any requirement to come to the University Medical Center. So we're doing all of the data collection in retirement homes, churches, and other areas, other centers within the community where we can rent space. Otherwise, it's a very parallel protocol to WITS. Another study that I wanted to mention is the study of Latinos, investigation of neurocognitive aging, or SOLINCA. So we have a study recently published with Dr. Stickle at SDSU Dr. Hector Gonzalez here at UCSD, looking at sex and reproductive history in Sol Inca and Latinos from across the country. And we found some really interesting findings. I encourage you to look it up. But we found specifically that oral contraceptive use is associated with less cognitive decline in this cohort. And we will be adding plasma tau markers into this data set very soon. So finally, I just wanted to spend a couple of minutes talking about how we might be able to help individuals to reduce their risk. So along with my colleagues Howard Feldman and Sarah Mukazel here at UCSD, I'm very frustrated by the fact that we know so much about modifiable risk, but when we see patients in the clinic, they often don't know that some of these factors are really associated with brain health. And they also don't know that there's a huge amount that they can do to reduce their risk. Even if they're at the beginning stages of cognitive decline, there's certainly things that they can do to slow things down or to reduce further decline. So we created the Healthy Actions and Lifestyles to Avoid Dementia study, or HALT-AD. And this program is used to educate people about modifiable risk and empower them to make lifestyle changes. So HALT-AD, or there's a Spanish version, Spanish language version that's been modified to be appropriate culturally for the local Latino population here in San Diego. And this educational program is delivered entirely online. And the idea is to provide really quality information about modifiable risk factors in a really digestible, kind of fun format. Because we know that education alone doesn't promote behavioral change, we accompany HALT-AD with a psychosocial support group, which helps to build community and helps to empower individuals to make changes and just keep them on track. So this is what some of the risk assessment component of the program looks like. People put in their individual risk factors, and as a result of that, they get a personalized learning program. And these are the modules that we cover. So of course we have an introduction, and then a diet, loneliness, sleep, physical activity, and so on modules, just to look at some of the data and to hopefully help people to understand the links between what we know helps and what you might do to make changes in your individual life. We help people to make SMART goals, so simple, break down sustainable changes in their own lives. So on that note, I want to thank you all for listening today. I want to thank the participants of all of these different studies that I've showed you, the people who lead the ADNI study, which we base a lot of our work on, our funding sources, including the National Institute on Aging, which funds our current studies, my 3 labs, the Human Memory Lab, the WITS Lab, and the HALT-AD group, and these other funding resources as well, who funded things in the past, California Department of Public Health, Alzheimer's Association, and the Women's Alzheimer's Movement. Thank you. And that's my contact information.
Sarah, that was wonderful. Thank you so much for such a comprehensive talk. And I have a couple questions.
Oh, sure. Go for it.
Question 1 is my lab participates in the Diane study.
Oh, yes.
And so dominantly inherited Alzheimer network. And of course, I'm at WashU and Dr. Bateman is here. But I was wondering if an inherited allele, which is aside from a risk score, right? So a risk score is going to look at perhaps associated alleles for an accumulated risk, and it's not just necessarily one. But in these families where there is obviously one allele that could convey a huge risk of Alzheimer, do you see as much sex difference?
I believe they have seen them. So Yakil Quiroz is very involved in that work, and she has looked at sex differences. there's a lot of things to think about, including that in these dominantly inherited Alzheimer's diseases, the onset of the disease is a lot earlier.
Mhm.
And it often coincides with the age when women tend to have menopause. So, whereas for Alzheimer's disease, the late onset, more common, thankfully, variant, you tend to have cognitive loss in your 70s or 80s. In diseases which are rare and dominantly inherited, the onset tends to be in the 50s, sometimes early 60s. And I believe, I'm not an expert in the early onset dementias, but I believe it is seen a little bit earlier in women. We do see parallel sex differences as we see in the later onset Alzheimer's. And there's some interesting work looking at how that might be associated with the actual onset of menopause, which of course affects inflammation, it affects sleep, it affects mood. It's sort of it's a big event for women, which has been largely ignored. And then another way that we're looking at that is in people with Down syndrome, who very sadly are almost always, if they live long enough, and these days generally do, thankfully, due to changes in cardiovascular health management, people with Down syndrome will also get Alzheimer's disease. And again, that tends to be earlier, I believe, in women than it is in men.
Interesting. I have had many patients with trisomy 21, so it's something I'm extremely familiar with. Um, and I guess, you know, I wanted to know, as my second question, how good you think the risk scores are. In other words, are there enough quantitative measures that can be added to a risk score such that it is going to really give you a good indication of risk in the future?
I think that's a really good question. And it speaks to whether or not we should be getting tested ourselves. What does it mean? Is it worth the stress of knowing? Because, if it's not going to happen and it's wrong, then why bother? And I think that also speaks to modifiable factors. So, genetics is a non-modifiable factor for the most part, unless you're doing
Mm-hmm.
some really snazzy manipulations. Um, so if you have two individuals, twins who are separated and raised differently, and one of them has low education and is overweight in midlife and has a poor diet and all of these different factors, and the other one has a great education and plays tennis every day and eats a vegetarian, super healthy diet, then those two individuals have different risk, even if they
Mm-hmm.
have exactly the same genetics.
Mm-hmm.
So I think it's important when we look at our genetics, and I advise people of this when they're saying, "Oh, I want to do 23andMe and find out what my ApoE is or whatever." Um, it's not everything, right?
Mm.
If you have one of those autosomal dominant, yes, it's probably very highly predictive of your outcome. But these risk factors, which are less certain, really interact with what we do and how fortunate we've been. So I think it's a complicated question, because they're explaining a fraction of the risk or a percentage of the risk, and that's kind of different for each individual.
Thank you. Um, I once had a patient who she had been my patient for quite some time before she did her 23andMe, and she was homozygous for the ApoE4 and literally became almost suicidal because she was sure it was going to convey Alzheimer's, not just maybe an increased risk. So
Right.
talking her off the ledge was something I did.
Oh, well, good.
um,
Thank you. And I think as well, some of those tests are not very accurate. ApoE
that's
is
another
actually
thing.
a hard gene to measure.
Yeah.
So there's different factors there where it's like it might not be right. And I know in some of the older direct-to-consumer studies, there was a lot of inaccuracy.
Mhm.
And even then, we're seeing now modifying diet, changing exercise, even if you're at very high risk, like someone with who's homozygous for ApoE4, it makes a difference. It can push back the disease for years. So, of course, we should all be doing those things. But if it's motivating to tie it to brain health and future brain health, then even better.
Wonderful. I agree completely on that one. Um, and I think we're going to close. I'm sure that I will have more questions, so I hope you don't mind if I email.
Please.
And this was really an excellent talk, and I thank you so much for your time and effort.
Thank you. It was wonderful to meet you, and thanks for the opportunity.